Good morning, everyone. Myself, Priyanka Kulkarni, Assistant Professor at Kekewa College of Agricultural Biotechnology, Nashik, in the Department of Microbial and Environmental Biotechnology. Today, we'll discuss the subject of basic genetics. So, the basically introduction part of the basic genetics. So, first of all, rather going directly for the basic genetic subject, so what is mean by the genetics, the very important thing is that we have to understand what is mean by the molecular biology, or what is mean by the central dogma of molecular biology, because the basic things or the basic concept which are the base of that genetics, they are coming from the microbiology. So this is very basic to understand what is the central dogma of molecular biology. Basically, it states that DNA contained the instructions for making the particular type of the protein. So, DNA means what? DNA means deoxyribonucleic acid. It is a one type of the genetic material which is present in organisms. And what does this central dogma of molecular biology states? It states that DNA, it contained the instruction or set of instruction or set of messages for making the particular type of the protein required in our body, or particularly it is required in that organisms. Yes. So in this process, the RNA molecule is also involved. So this is a basically short pathway or short cycle of conversion of DNA to the RNA and RNA to the protein. So RNA, we can call it as a mediator. Okay. Yes. And this cycle or this short cycle is known as the central dogma of molecular biology. So DNA means deoxyribonucleic acid, which is a genetic material. It is present in each and every organisms. Okay. So the copy of itself means making the copy of itself. That process is known as the replication. Replication means producing the replica of itself. Replica means copy. So producing the replica of itself means DNA produces the replica of itself. That is the process known as the replication. Means from the one DNA molecule, the two different kinds of the DNA molecules are produced. So that process is known as the replication. Then next process is known as the transcription. Transcription, it is a process in which the information which is present on the DNA, double-stranded DNA, it is converted to the single-stranded RNA, or basically the mRNA. So the information is transcribed to the mRNA, and then that information is translated to the protein. Translated means what? Means translation process, means conversion of RNA to the protein. Means here the language is changing. The language is changing from the nucleotide to the amino acid sequence. That's why the word translation is used over here. Translation means the translators, we are generally, there are so many translators to translate the different kinds of the language from the one language to the another language. So that person which actually translate this language, we call that person as a translator. Okay, so here also the same thing is going on. Here also the translation process is going on from the language of the nucleotide to the amino acids. Means RNA, from the RNA the protein is producing. Okay, so that process is known as the translation. And there is a one more event that is known as the reverse transcription. Reverse transcription means what? It is the opposite of the transcription. Okay, it is basically used to prepare the cDNA library or complementary DNA library. So RNA means whatever the mRNA which is produced after the transcription process, that mRNA again reverse transcribed to the DNA to produce the complementary DNA molecule. Okay, so this short event or this short cycle is known as central dogma of molecular biology. Means your total subject of molecular biology is running between these things: replication, transcription, reverse transcription, and the translation. So what is the basic bottom line of this slide is, from the DNA, RNA molecule is produced, and from the RNA, protein molecule is produced. So basically, whatever the information which is required to synthesize the particular protein in our body, that information it carries by the DNA, which is present as a genetic material in the organisms. Okay, so this is all about the central dogma of molecular biology. Then next, we'll go for our subject, that is the genetics. Okay, so what is mean by the genetics, or what is basically genetics? Genetics, it is a branch of biology which deals with the heredity and the variation of inherited characters. Okay, easy definition: it is a branch of biology which deals with the study of heredity and the inherited variations among the inherited characters. Okay, so what is mean by here heredity and variation? Heredity means there is a something, or there is a genetic material which is transferring from one generation to the another generation, or from your grandparents to your parents, from your parents to you. So that genetic material or that flow is known as the heredity. Okay, yes. And variations of inherited character. Variations means what? Still there are genetic material inherited from your parents to you, your grandparents to you. Still there is a difference between the you and your grandparents and you and your parents, right? So that variations we also have to study, and that variation is of what? That variations are of the inherited characteristics. Means whatever the characteristics which are carrying, we are carrying from our parents. Okay, inherited means what? Which are inherited means which are transferred from our parents to us. Okay, so that flow is known as the heredity or inherited characteristics. Yes, so here one figure is cited. You can able to see that there is a daddy mouse. Okay, daddy mouse whose chromosomes are given. Daddy mouse is a heterozygous. Okay, why? Because its one allelic form is dominant and one allelic form is recessive. Okay, so this daddy mouse is heterozygous for this B. Okay, B we can take as a black color of the eye. Okay, so here at least one character is capital, or at least one character is dominant over here. So the eye color of the daddy mouse will become the black color. Why? Because the black color dominant gene or dominant allele is present in the chromosome of that daddy's mouse. Okay, so the same genetic material is transferred from daddy to offspring, right? And one genetic material is also transferring from mother to offspring. Okay, so suppose from the daddy, only this chromosome, only the small b chromosome is transferred to this baby. Okay, and from the mother also the small b chromosome is transferred to the baby. Okay, so what would be the baby's eye color? Both are in a small bb. Okay, means both are the homozygous, right? Both are the homozygous. Okay, and both are the small, means both are the recessive. Okay, so what is the recessive color of eye? It is a pink or red color. Okay, so that the baby mouse will develop the pink or red color of the eye. Okay, So here in this figure, you can see both heredity and variation among the inherited characteristics. Okay. So heredity means what? The eye color gene or eye color allele is transferred from the mother and father to the baby child. Okay. But the baby child will develop the eye color which is depend upon the combination of the both. Okay, which is depend upon the combination of the mother and father. Okay, yes. So this is the variation, means still this is transferring to baby, but still the eye color of baby is different and eye color of father is different. So that is called as the variation of inherited characters. Okay, and the gene which is transferring from mother to the offspring, that is known as the heredity. Okay, so that's all we have to study in the subject of the genetics. Okay, so genetics, it is a branch of the biology which deals with the study of heredity and the variation among the inherited characteristics. Okay, so this is basically your genetics subject. Okay. Yes. The next one is introduction. Okay, the same thing is given, that what is the definition of the genetics. There are so many types of the genetics definition, you can go and search, which is put forth by the so many different kinds of the scientists. Okay, but we will just go for the simplest one. So what is the simplest one definition? Definition is, it is a branch of the biology which deals with the study of heredity and the variation among the inherited characteristics. Okay, yes, it is also a study of genes, genetic variations, and the heredity in the living organism. This also can be the definition. Then, genetics, it is a study of inheritance in all of its manifestations, from the distributions of the human traits in the family pedigree to the biochemistry of the genetic material in our chromosomal DNA. Okay, so this is somewhat lengthy definition. You can go for the small vowel definition. Yes. Then before going for the actual genetic subjects, there are so many terminologies or so many definitions are there. You need to understand that definitions. Okay. So we'll go for one by one definitions. So gene. So genetics, the word, it is made up of from the gene. Okay. So what is mean by the gene? Gene is a biological unit of heredity. Gene is what? It is a biological unit of heredity, or it is a physical, functional, or biological unit of heredity. Not or and. Okay. It is a physical, functional, and biological unit of heredity. So, gene is what? Gene is nothing but only the small segment of the DNA. Okay, gene is what? It is a small segment of the DNA. So, what is the function of this? Its function is it's hold the information to build a particular kind of the protein. Okay, as we seen in the central dogma of molecular biology, okay, we have seen now that DNA converts to the RNA, RNA to the protein. Okay, so the same thing is happening over here. DNA means what? DNA means short segment of the DNA is called as the gene. Okay, or gene is what? Short segment of the DNA. So that also carries the information to produce the particular kinds of the proteins. Okay, so that kinds of the proteins are produced from the gene, or gene carries a particular message to produce a particular kind of the protein. So what is the basic definition of the gene? The basic definition is, it is a basic physical, biological, and the functional unit of heredity. Okay, yes. Then there are the some genes which act as a means which carries the information to produce the protein, but there are some genes are also present which does not carry any information. Okay, they are the useless genes which are present in our body. Okay, yes. So there are two different naming system, introns and the exons, exons. Okay, exon means what? Exon means the gene segment or the gene which is uh, which provide the particular information to synthesize the protein, that is known as the exons. And introns means what? Introns means these are the gene segment which does not code for the anything, means these are the useless gene segments. Okay, yes. So this is all about the genes. Okay, basically in the humans, genes are varies in the size from the few hundred bases to the more than 2 million kinds of the bases. Okay, so the length of the genes are very, means length of the gene, it is not customized, or it is not what we can say, fixed. Okay, so length of the gene is vary from its gene to gene. Okay, some genes are very small, of only 10 to 15 bases or only 100 bases, but some genes are very large in size, like they are of the millions of the bases. Okay, yes. The flow is like this. Gene means small segment of the DNA. Okay, convert to the RNA, it converts to the protein. And what is the function of the protein? Proteins are required for your body metabolism. Okay, to maintain the functions of your cell, to create the different kinds of the enzymes, to produce a particular kind of the cell structure. These are all the functions of the proteins. Okay. Yes. So we will stop here. We will discuss the next point in the next lecture.